Assalamu alaikum. Today we are going to discuss the quality maintenance of seed bank technology. Quality maintenance of seed bank technology. So, what is now seed bank? We can say it is the conservation of or preservation of those species which are commercially valuable or which are at the verge of extinction. The currently extinction rate of plant and animals has been observed 100 to 1000 folds higher due to their destruction of natural habitat. So, if there is a destruction of natural habitat, so these plants are not naturally available. So, it is responsibility of us. It is responsibility to us that we have to conserve these seeds or these plants under in situ conservation or conservation outside their natural habitat. So seed bank, in case of seed bank, we have to store seeds under cold and dry conditions. But it has been observed, while keeping these seeds for longer time for future use, they loses its viability. They loses its viability. So in US they are having many national plant germplasm systems where they are able to store domestic as well as non-domestic species. They have maintained botanical gardens as a conservative source for these plants. So in Britain, they have made seed bank project at the Royal Botanic Gardens, Kew in Great Britain, where they have stored at least 10% of world's plant diversity, and mostly they have targeted to dry topics as well as the species which are native to the Great Britain. So in Britain they have also maintained their they have conserved their plants which are native to them. In USA it has calculated that 700 botanical gardens maintain these seeds, or they maintain wild types of plants, they maintain ornamental medicinal plants as well as crop or crop related species in those botanical gardens. So we can say that we have to conserve wild plants, wild plant species. Either the species are related to our crops, or it's related to our pasture species, trees, or medicinal plants. Day by day, these seed bank conservation is getting popularity, which has contributed a lot to the society. As in seed bank, we are having immediate access to the seed bank. Second, we can say that the plant germplasm is available anytime, which help us or which facilitate us in the scientific studies. And the seeds which are present in the seed bank are immune to their habitat destruction, because we have taken these seeds from their natural habitat only, so they are well adapted to the environment. If that environment is hazardous, or if there is climatic change in that environment, so these plants or trees or the species or the seeds are also habitual of those destructions or those changes as well. So we can say that diversity is there. There is small genetical modification with respect to its environment. So if there is a, even nowadays it has been observed that the environment has shifted dramatically with respect to the presence of water, increase in the temperature, or related to its natural habitat. So when there are these under these circumstances, the gene frequency and the gene combination of the genes has changed in the plant, has changed their gene combination of genes, so that such combinations are available in in these plants. They will withstand with the high temperature or withstand with the low availability of water under the natural conditions. But due to these seed bank collections, these allow us to store and regenerate outside their natural habitat, typically in such a way as to minimize the genetic change. So we can say about that we are having many factors which are responsible, which affects our seed bank. First is the adaptation and genetic shift accompanying regeneration in seed banks. As we are regenerating, when we have maintained our seed in a seed bank under artificial conditions, we allow it to regenerate. So these plants under in vitro condition are able to grow in a similar type of medium, or similar environmental conditions are available there, which maintains these, which allow these plants to grow in a very similar type of conditions. But under natural condition, when we was allowing these seeds to grow under natural condition, they were having, or they were facing different environmental conditions, due to which there were the genetic variation. But due to this in vitro culturing of the seeds, they have lost their ability, they will lose their ability to grow under the natural habitat. So we can say that this contributes to a shift in the genetic composition of the plants, or genetic composition of the seeds. Even in breeders, even in plant breeding program, breeders consider that contamination with the pollen from the adjacent plant is also the destruction of the pure line, as we have discussed this in the previous lecture. So the next factor is loss of viable variation by genetic drift. Loss of variation by genetic drift. Collecting and regenerating seed samples is laborious, and it's not possible to collect and preserve all the seeds available for a particular species. We can collect few of them, like 100 individuals in a wild, but while storing them, they lose their adaptability to the natural environment. So we can say that it leads to the loss of genetic variation that may contribute to the increased probability of extinction in the wild species. When these seeds under seed bank technology are not grown under the natural conditions, so under natural conditions they will change, drastic change in the environment, due to which they will be able to adapt the variations. But if we are growing these seeds under in vitro condition, similar type of procedure, similar type of environment is providing to them, so they may lead to probability of extinction. So due to which the wild population can lead to loss of genetic variation due to the genetic drift. It is also well known that the influence of this genetic drift on variation at a single locus depends on the size of the collection, means how much we have sample taken, and the frequency distribution of alleles in the genome, and the frequency distribution of alleles in the genome. The another factor we can discuss is the inbreeding depression. So when we have collected the seed, we have to regenerate it. Due to this repeated regeneration, Due to this, repeated regeneration of these seeds under in vitro condition increases the degree of relatedness, and these seeds are prone to the low germination. When there is a regeneration is when we are allowing it to regenerate again and again, at last these seeds then are facing problem in their germination. So these seeds at the last they finally lose their germination power. So this is the inbreeding depression. So even there are some mutations when we keep these seeds for longer time, or when we store these seeds for longer time, that is the aged seeds. It has been seen that in aging seeds undergo increased rate of mutation. So we have to take care that while we are preserving these seeds, conserving these seeds under seed bank technology, so we have to be sure that there is no such mutations which may be lethal to the plant, or we may lose the whole seed bank for that particular crop. So it is always recommended that we have to go for a controlled experiment, but that experiments had never happened with the seed bank materials. Honestly, that technique is there, but they have never carried out experiment to these seed bank materials. Seed bank in the service of ex situ conservation efforts. So earlier when they have started for seed bank, they used to conserve agriculture seeds only, but later on they start conserving, they extend to the wild species as well. So this preservation under access to conservation is helpful for the human society, and by conserving these plants or the wild species, while conserving these wild species under in vitro condition or in seed bank technology, whenever we are in need of these wild species, we can take wild species, we can isolate some particular gene from them, we can get variation, we can get hybrid plants from these wild types of species. So it is important for us to store plant in their wild form, in their native form. So when we store these plants under native form, then we will be able to get novel genes for gene transfer from these wild type of plant species only. So we can say that seed bank can be maintained or conserved in botanical gardens as well. And we have to make public aware that we have to conserve our nature. We have to save seeds at the global level, as there is the one organization that is called as Savalbord Global Seed Wallet. So this seed wallet has made one community seed bank. This community seed bank works at the local level, which help farmers to assess the growing crops, which allow farmers to assess their seeds to the grown crops, or at the time of emergency when there is damage or destruction to the crop due to the climatic change or due to the agriculture misfault in the agriculture production. Growing local varieties will have a higher degree. By growing these local varieties in a particular community, will be having a higher degree of genetic variation. मीन्स एक पर्टिकुलर एनवायरनमेंट है हमारे पास, वहाँ पे क्लाइमेट चेंज हो रहा है, तो जो प्लांट उसमें सर्वाइव कर रहे हैं उनमें एडेप्टेशन आ गई है. ड्यू टू एडेप्टेशन जो उनमें आ गई है, डैट इज़ ड्यू टू देयर जेनेटिक डाइवर्सिटी. Due to presence of this genetic diversity in their genome, these plants are able to survive the climatic change. So we can say that community seed bank is also useful for the community. We store our seeds. These seeds are directly taken from the farmers. So in a single community, we can store these seeds in pots, or in, if we are having some community buildings, or we can store these clay pots on the floor, even on the shelves of the kitchen. Whenever there is need of these seeds in the community, these seeds are provided to the people in a particular community. So commercial or new varieties coming from the national breeding program are usually created by the local varieties. Whenever we have to go for a new variety, we always prefer to take local variety, then we interbreed these two varieties, then we will be able to get a new variety, as we have seen in the hybrid wheat in our previous last lecture. So this was about the how we will be able to store or maintain our seed bank technology. The next is the growing our seeds or growing our plants or plant plants under the minimum growth storage. So in case of minimum growth storage, what we do, we don't allow plants to grow under in vitro conditions. We only allow it to survive under in vitro conditions, that is minimum rate of growth are allowed. So this type of preservation or technique is mostly available to the plants from the explant, which includes our shoot tip cultures or meristem derived plantlets or meristem derived plants. So with the minimum growth storage, we can change the physical condition of cultures that can be altered, like if we will change the temperature or the gas composition within the culture vessel, or we can change the basal medium, or the medium can be altered by giving low amount of carbon source in the medium, or we can go for a suboptimal level concentration of other nutrients as well. The medium can be supplemented with growth retarded, like if we will have a medium MS medium, in this MS medium we are adding abscisic acid or osmoregulatory compounds such as mannitol or sorbitol, which will create osmotic pressure on the surface of the explant, and this explant will not be able to grow much. It will survive, it will remain alive for a longer period of time. And the last of this is the cryopreservation. As we are familiar with this cryopreservation, that we have to store our seeds in with the cryoprotectant in the liquid nitrogen at minus 196 degrees centigrade. So the steps which are used for cryopreservation, we can't directly take our plant and directly immerse in the liquid nitrogen. No, whatever the plant or the seeds we have to store, it should be specific for us whether they are tropical plants. We have to be sure that whether they are tropical plants or the temperate plants. If they are temperate plants, they will respond good to the cold conditions. If the seeds are taken from the tropical plant, giving directly their cold treatment at minus 196 degrees centigrade, these tropical plants will not survive or will not remain live for longer period of time. So start with the steps in the cryopreservation, we have to go for a pre-treatment. We have to go for a pre-treatment specific for the tropical plants, specific for the tropical plant. We will take a seeds, we will allow it to remain in a we will allow it to improve its resistance to cold. How we will improve its resistance to cold? By giving long cold nights and short warm days. By this treatment, scientists have observed that the plants which are from tropical region will be able to survive by giving long cold nights and warm short warm days. So when this condition is maintained under these conditions, when plant will get adjusted, then only we will proceed for a next step. When the seeds are now acclimatized to this condition, then we add to these seeds we add cryoprotectant. We add cryoprotectant. We are having many number of cryoprotectants used for in vitro techniques. We are having one. DMSO is the most common. The full form of DMSO is the, we can use 5% DMSO, dimethyl sulfoxide. So dimethyl as a cryoprotectant is used with the concentration of 5%. We are having another cryoprotectant, that is DGS. In DGS cryoprotectant, we are having combination of three components: 1 molar DMSO, 1 molar glycerol, and 2 molar sucrose. Or we can use as a cryoprotectant, we can use sucrose with the concentration of 0.75 molar sucrose. And this sucrose as a cryoprotectant can be used for cell cultures or shoot tip cultures. There are many techniques which are involved in the cryopreservation. Cooling, we can go for slow down, or we can say that we have to give the stepwise cooling or slow cooling. This is one of the techniques. What we do here, we take X plant, then we allow starting temperature, we allow it to grow under less than 1 degree centigrade. When they are there for 1 degree, then with gradual we are decreasing 1 degree centigrade, that is less than 1 degree centigrade per minute to -40 डिग्री सेंटीग्रेड पर मिनट. मीन्स हम लोग एक मिनट में एक डिग्री कम कर रहे हैं, स्टार्टिंग फ्रॉम द 0 degree और 1 degree till we will reach to -40 degree centigrade. मीन्स ग्रेजुअली स्लोली हम लोग क्या कर रहे हैं, हम लोग एक डिग्री कम कर रहे हैं, माइनस में जा रहे हैं. When we reach at the -40 degree centigrade, then we directly take these seeds along with the cryoprotector, then place directly into the liquid nitrogen, which is having the temperature of -196 degree centigrade. Another technique we can use as a cryopreservation is rapid cooling. We directly take our sample, add cryoprotectant in it, then directly immerse this into the liquid nitrogen. The third technique which are used for the cryopreservation is the encapsulation dehydration. As the name indicates, encapsulation, we have to encode these seeds with agarose or the alginate. Then we have to go for dehydration process. So this dehydration, after dehydration, we have to then immerse these seeds into the cryoprotectant, then into the liquid nitrogen. So these were the different techniques which were used for the preservation of the germplasm or the seed bank conservation.